Hi everyone, I'm Frederic Pontvianne. I'm a CNRS researcher in the Plant Genome and Development Laboratory located in Perpignan in the south of France. And I will talk to you about the functions of the nucleolus. So the nucleolus or nucleoli were actually first described a long time ago by Fontana in 1781 after the first development of the light microscope. And it's after the great work actually of two great cytologists, Heitz and Barbara McClintock in 1934, while she was still a young researcher, that a link between the structures, the nucleolus, and chromosomal secondary constructions that are named now NAD, for nucleolar organizer regions, was made. And actually this name, nucleolar organizer region, was found by Barbara McClintock herself. We now know that this secondary constriction correspond to the location of ribosomal RNA genes that are organized as tandem repeats in this area. Each unit corresponds to an intergenic spacer and a transcribed region by the RNA polymerase 1, which is going to transcribe the long precursor 45S, composed by 18S, 5.8S, and 25S. And after this transcription, there is a lot of different steps of maturation and processing to give rise of the pre-ribosome 40S subunits and 60S subunits. These steps of ribosome biogenesis, and most of these steps are located in the nucleolus, but some late processing steps happens in the nucleoplasm and in the cytoplasm. Interestingly, if you look at the structure of the nucleolus by electron microscopy, you can also find subnucleolar structures that actually correspond to steps of ribosome biogenesis. The clear area are fibrillar center, surrounded by dense fibrillar component, and everything is embedded in what we call granular component. This structure can also be revealed by confocal microscopy if you fuse, for example, fibrillarin 2, which is a protein implicated in the dense fibrillar component in several processing steps. And then if you fuse it to a fluorescent protein, you can reveal the presence of this ring-like shape corresponding to the DFC. So as I said, these structures are actually closely linked to the subnucleolar compartments. The more you're in the center of the fibrillar center, for example, more you're in early steps of ribosome biogenesis, which start by RNA polymerase 1 transcription of ribosomal genes precursor. And then you're in later step, you are in a dense fibrillar component and then in a granular component. This is called the concentric functional organization. But the nucleolus is also implicated in other functions, in RNA maturation, RNA turnover, genome nuclear organization, stress responses, for example, and many others actually. And to identify and to reveal these different functions, one way is to isolate nucleoli and identify its content in protein, in RNA, or in nucleolar DNA. In our lab, we actually developed a way to purify nucleoli using fluorescent-assisted cell sorting. And for that, we use a marker, fibrillarin 2, fused to a yellow fluorescent protein. And we can use the fluorescent of the fibrillarin 2 or DAPI to directly isolate nuclei by cell sorting, or after a sonication step, we disrupt nuclear membrane, and the nucleoli are going to stay intact because they are very dense structures. And we can use the fibrillarin 2 YFP signals to sort them by fax. I will briefly focus on the role of the nucleolin in genome nuclear organizations in the context of the, which is basically identification of nucleolar DNA. Indeed, in the context of the in-depth ECOST network, we are interested in how nuclear domains impact gene expression. And actually, at the nucleolar periphery, there is chromatin domains that are features of repressive features and are more like heterochromatin, like at the nuclear periphery. So these specific chromatin regions are called NADs for Nucleolus-Associated chromatin domains. Indeed, if you look at a dapistane nucleus from Arabidopsis thaliana, you will see the nucleolus like a black hole. So there is not a lot of DNA, but if you start to isolate them, you can reveal the presence of the DNA. If we now take Arabidopsis thaliana, we can isolate nuclei or nucleoli, isolate their content in DNA, and identify Nucleolus-associated chromatin domains. That's what we did, and we did not find only ribosomal RNA genes. On this dot on the left, you can see all 5 chromosomes of Arabidopsis thaliana, and the more you enrich in a nucleolus, the more you're on the top of the curve, and you become red. If you if you identified as NADs. If you look at the distribution of these NADs, you can first see a large region on the short arm of chromosome 4, which is enriched with the nucleolus, and which is just next to the NOR4. So NOR4 actually correspond to ribosomal RNA genes that are actively associated with the nucleolus and expressed, transcribed by RNA polymerase 1, while there is other NOR2 derived ribosomal RNA genes, but they are excluded from the nucleolus area, and we don't see any enrichment in that case. In addition to the short arm of chromosome 4, we also have a smiley shape, which basically means that the end of chromosomes associate with the nucleoli. This corresponds to telomeric and subtelomeric regions, and indeed, some years ago, Susan Armstrong works, as well as Paul France, revealed that telomeres tend to cluster in the nucleolus. We also have additional regions, and if we take all of them, we can see that they are enriched in transposable elements and depleted in genes. However, we still find 900 genes present in this area. Although there is no specific categories, we can find enrichment of pseudogenes and tRNA genes. So globally, these NADs, all these regions, they display heterochromatic features, including CG methylation and H3K9 demethylation. Interestingly, if you look at RNA Pol2 immunolocalization, you can see that it's basically present in the nucleoplasm, but it's excluded from the nucleolus area. And indeed, if you look at global gene expression of genes present in these nucleolus-associated domains, they tend to be low expressed. So there is potentially a regulation, a role of the nucleolus in gene regulation by potentially sequestration. So if we come back to our global genome organization, we have chromatin domains with heterochromatic features at the nucleolus periphery, as well as at the nuclear periphery. And if you combine both to see the overlap between them, you can see there is actually very few overlap between NAD genes and LADs for lamina-associated domain at the nuclear periphery genes. So take-home message is that the nucleolus is the largest nuclear compartment. It's a site of ribosome biogenesis. It is also implicated in other functions, like nuclear organization. And if we look at the nuclear organization as a library, we also have like storage area at the periphery or in the nucleoplasm. which can be seen like closed book, and reading area where information is accessible only if you open the book. And basically the nucleolus has also a storage function, but really has a specific function in the middle of the library. Thank you for your attention. And if you need more information, please look at the following references.